We are continuing the glycolysis process, the second part. We have already done the first part, and it's in part one video. So if you have not watched that, just go and watch that, because this is only the second half of the information. The first half is on the first video. So yes, definitely go and check that out. Here, let's just get started now. We left off in this particular molecule in our previous video, glyceraldehyde 3-phosphate. The next step is to convert that to something called 1,3-bisphosphoglycerate. And as I have mentioned before, you see this kind of name, you can figure out a lot of things from here. We have phosphate and number 1, phosphate and number 3. The bis tells us we have 2 phosphate. And the enzyme involved is glyceraldehyde 3-phosphate dehydrogenase, GAPDH, that's the short form. And this is basically a redox reaction. In redox reaction, we have reduction and oxidation both happening at the same time. So this is where the reduction is happened. The reduction of NAD plus to NADH. NADH is one of our products of glycolysis, and this is where the oxidation happened in this process. Next step is converting that 1,3-bisphosphoglycerate to 3-phosphoglycerate. As you can notice, there is no 1 here. So what happened to that phosphate is that phosphate basically got converted to ATP. So we have ADP. So the phosphate from this particular phosphate went to ADP, and that become ATP. There you go. And this entire process was successful because of this enzyme phosphoglycerate kinase. As I mentioned before, you see the word kinase. It's basically attaches one the phosphate from one group into another, and this is a single-headed arrow. So going from 1,3-bisphosphoglycerate to 3-phosphoglycerate and ATP is an irreversible reaction. Whenever ATP is involved in the reaction, it's usually going to be a single-headed arrow. And this is a double-headed arrow. You can go back and forth depending on the situation. And this is also our first substrate level phosphorylation. We made ATP. That was the purpose of glycolysis, to make ATP, to make NADH. So here we make our first ATP. And the next step is to go from 3-phosphoglycerate to 2-phosphoglycerate, and that is basically just a movement of phosphate from third carbon to second carbon. Nothing else happened here, as you can see. And we have this enzyme phosphoglycerate mutase. This is the enzyme that basically rearranges the location of phosphate, in this case, going from 3-phosphoglycerate to 2-phosphoglycerate. The next step is to go from 2-phosphoglycerate to phosphoenol pyruvate, and that is a condensation reaction. The enzyme involved is enolase, and whenever we have condensation reaction, we are going to see water, and just like that, we have water as a product in this reaction. Let's go. This is also a double-headed arrow. And the next step is to go from phosphoenol pyruvate to pyruvate. So as you can see that that phospho part is missing. So what happened is that phosphate, again we have ADP, we have ADP, we have ADP, that phosphate came to that ADP and became ATP, and this entire process converting from phosphoanyl pyruvate to pyruvate was successful because of this enzyme pyruvate kinase. Again, kinase, you see the word kinase involves with the movement of phosphate. It attaches the phosphate from one group into another. That's what pyruvate does. And this is also a one-headed arrow. So this is also our substrate level phosphorylation. We made 2 ATPs. So one we made right here, and the other one we made right here. These are the two steps in where a substrate level phosphorylation happens, and we made 1 NADH. However, don't forget that when we started glycolysis, the glucose was 6 carbon sugar, and we converted that into 2 3 carbon sugar. So this entire process, this entire cycle happens twice. So we make 1 ATP plus another ATP here, and we make 1 ATP plus another ATP, total of 4 ATP and total of 2 NADH. So that gives us total of 4 ATP and 2 NADH for the entire process of glycolysis. Let's just summarize that actually. In summary, we have one molecule of glucose, which is a 6 carbon. That's a 6 carbon sugar, and that is converted to, in one cycle we have pyruvate, pyruvate, we have pyruvate, we have ATP, and we have NADH and ADH. So we have this process happens twice. So we have 2 pyruvate, we have 4 ATP, and we have 2 NADH. However, we also used 2 ATP as an investment, minus 2 ATP. So the overall is, our net ATP is 2 ATP. 2 ATP. Okay, yeah, that's the entire process of glycolysis. We have one molecule of glucose that is converted to 2 pyruvate, which is involved later in the process in TCA cycle, and we make 2 net ATP and 2 NADH, which we can use in electron transport chain to make more ATPs. All right, so let me give you one question to think. So what do you think will happen if we increase the concentration of ADP in this process, going from phosphoanyl pyruvate to pyruvate? What do you think will happen? So write your answers in the comment section below, guys, and I'll see you in next video.